Hi, guys. If you weren't in class, we reviewed protein synthesis and mutations. So here's a recap of what we did in class. First of all, protein synthesis is the way that your body expresses your genetics or your DNA. So we take our DNA, we transcribe it into mRNA through the process of translation. So we copy DNA into mRNA. And then that mRNA is used in translation to build a protein. So that's the overview of how we do protein synthesis, the building of a protein. So part one is transcription, to make a copy of your DNA and turn it into mRNA. So transcription takes place in the nucleus of a cell. It's when DNA is opened up and a small portion of DNA, just a gene, is then copied into mRNA, messenger RNA. Now, an enzyme, of course, enzymes always help, but an enzyme is going to help with this process. And the reason for this is that the DNA is trapped in our nucleus. It's too large and too important to leave the nucleus. So the mRNA is going to allow that message to be taken out of the nucleus and brought into the cytoplasm where the protein can be assembled. So, if you remember in transcription, we use DNA as a template, but we're building mRNA. So, you might be asked to look at a DNA molecule and tell me what mRNA molecule I would make. And so, in this case, you need to remember the apples under the tree, cars in the garage acronym. So, when there's an A in DNA, it used to pair with a T, but there are no T's in mRNA. So instead of a T, I use a U, a uracil. G still pairs with C. That's the easy one. So what do we do with the T that's in DNA? Well, T still pairs with A, and that's okay because there are A's in mRNA. So every time we see a T, we put an A. Both C's become G's, G's become C's, T's become A's. And the exception is when we see an A, we wanted to put a T, but we have to put a U. So let's look at this example. If we were building the other side of DNA, G would become C, T A, C to G, A to T, G to C, A to T, A to T, A to T. We could go on all day. But we've already practiced those complementary bases with DNA. Apples in the tree, cars in the garage. So what you really need to focus on is the difference between taking DNA to DNA and taking DNA to mRNA, which is what transcription is. So that's where you need to worry because you don't have the use of T's. So C's still become G's, but instead of a T, we're going to put a U for mRNA. G's become C's, T's still become A. And so you fill out your mRNA using the mRNA complementary bases. All right, once you've assembled your mRNA, then you can do translation. You can actually read it and make a protein. And so this is going to happen in the ribosomes. And they're going to read the mRNA and build the protein. So we will see lots of pieces. We'll see mRNA, which is the message of DNA, the ribosomes that do the reading, and the tRNA are special molecules of RNA that actually bring the amino acids to the ribosomes so that we can actually build a protein. Because remember, proteins are they're made up of amino acid. And so amino acids need to be brought to the ribosomes and put together to make the protein. So, to do this, we read our mRNA, the message that left the DNA, and we read it in three-letter groupings called codons. So, you can see here we pair 1, 2, 3, that's one codon, 1, 2, 3, that's two codons, and so on and so forth. What's really important is that you remember how to read a codon chart. And I'm going to tell you on the end of course state exam, it looks like they always give you the square codon chart. So you need to become proficient at how to use this chart. So what you're going to do is you're going to find you have your 3 letters in your codon. You're going to take the first letter and find it on the left. Your second letter you're going to find on the top. And your third letter you're going to find on the right. So let's just do U to be easy. So my first letter is a U. That's going to limit me to this top row. So my answer is in this top row somewhere. My second letter is a U, which means it's limiting to this column. But the only place those two intercept is this box. So now I've narrowed this down to this box here. Well, the last letter was a U. So that's going to tell me that I'm finding my answer in the top row. So if I were to see the codon UUU, I would need the amino acid PHE. So let's try that again. Let's say I have C as my first letter. That limits it to this row. If C is my second letter, it limits it to this box where they intercept. And then if my last letter is C, then that would give me proline, PRO, as my amino acid. We'll try one other one. Let's try UCA. So U limits me to the first row. C limits me to the second column. So U and C, that gives me this box here. And then the last one I said was A. So that's going to give me SER, serine. Now, this chart is really nice because it has all 3 letters in the boxes. But guys, the charts on the standardized test are not going to have these the 3 codons in here or the 4 codons in here. It's only going to give you the amino acid. So you really need to know how this chart actually works so that you can use it when you're taking your state test. All right, so let's do the translation part. So if I've already transcribed my DNA into my mRNA, I have G, UC, AGA, AAC, and CUG. And so now I need to find my amino acids. So on my chart, I'm going to go to G, I'm going to go to the column for U, and then the line for C, and that answer would be VAL. For the second one, I'm going to go to A, go to the column for G, and then the last one is A, so that would be ARG. The third one would be AAC, so go to the row for A, the column for A, and the last one is C, so that would be ASN. So here's my last letter, C. And lastly, I go to the row for C, the column for U, and then lastly, the row for G, and that's LEU. So I'm able to take my mRNA and turn it into amino acids during the process of translation. And that's the beginning of your protein. You would have thousands of amino acids hooked together, and then when you're done creating it, it's going to fold up into a protein with a very specific shape, allowing it to do its job. So, the next thing we need to talk about are mutations, things that can go wrong, and how that affects protein synthesis. So, a DNA mutation is any change in the nitrogen based sequence of DNA. So, changing one of the letters in DNA. And remember, mutations are only passed to the next generation if they're found in a gamete, which is a sex cell, a sperm, or an egg. And so let's take a look at the different types of mutations that we can have. A substitution mutation is the easiest mutation that you can have. It's simply when one letter is substituted out for another letter. So in this case, the T that was in the original DNA was subbed out for a C. So there can be 3 different effects of substitution. Sometimes you can change a letter, like in this case, my original DNA was A, but I changed it to a G. But you can see when I went through protein synthesis, I still got the same amino acid because the majority of the time, substitutions make no difference. They have no effect on the outcome of the protein. The second situation is that I could have changed that A to maybe a C. And you'll see in this case, when I went through transcription and translation, I got the amino acid TRP. But that's only one difference. So there's going to be one different amino acid. So this might not have a huge impact on my protein. It might not make any difference at all because it's just one amino acid out of thousands. The only way substitutions really make a big impact is if we accidentally substitute a letter that creates an early stop. So in the last situation, I only changed one letter, but I ended up with a stop. So that would stop the production of that protein and would, in fact, ruin it. So this would be a substitution mutation that would actually cause a huge difference in my protein. Most of the time, they cause little to no problems for the protein. The type of mutation that you really need to worry about is a frame shift mutation. These are when you accidentally insert an extra letter or delete a letter that was supposed to be there. So in this case, I take out the C or and everything this is an insertion. I insert this C and I bump everything over to the right, one letter. And so that's my frame shift is I'm shifting everything over one letter. In this situation, I'm taking out an A and I'm shifting everything over to the left to make up for that spot because we have to fill that spot in. So everything's going to move over to fill that spot in. But remember, we read in 3-letter words. So if things are moving to the left or the right, it's changing those 3-letter words, those codons. And so it makes a huge difference. It affects that codon. So it changes that amino acid and every single codon and every single amino acid that comes after it. So these frame shift insertions and deletions, these frame shift mutations have a huge impact on your outcome. To show you this, I did a sentence where I wrote in 3-letter words, the cat ate the rat. And if I just insert an extra letter and I shift every letter over just one, it becomes a pretty unlegible sentence. That, eka, ta, etha, ert. In fact, I added a base. So you can see at the end, I have one extra base that's just hanging out there. And that shows you that it was an insertion. In the second one, I did a deletion. So I deleted the letter E and I shifted all my letters to the left to make up for it. And I get thuk, atta, tet, her, at. Notice I have 2 extra bases at the end, and that's because I'm missing one that would have made up that last base because I deleted it. And so you can see that this messes everything up. It messes the codons up, so it messes the amino acids up, which will mess up the protein. So again, insertions and deletions are really bad mutations because they frame shift and they destroy the protein. Substitutions, they're really small change that may or may not have a big impact on your outcome. So, most people think instantly that mutations are all bad, but not all mutations are bad. In fact, when we looked at evolution, any new trait in a population comes from mutations. So, new traits don't just appear out of nowhere. A mutation had to happen in order for that to become a new gene. So, if a butterfly suddenly had red color, well, that must have been a mutation in their DNA. So, there are a few types of questions that you should be familiar with when you are going to answer things on the EOC. So, here are 2 released questions. In this first question, it's asking you what organisms are going to be able to pass the mutations onto their offspring. Well, here are the organisms L, M, N, and O. And they found mutations in the skin cell of M, the stomach cells of N and O, and the gametes of L and M. But guys, the only mutations that can be passed on are mutations that are found in the gametes, which are your sex cells, your sperm or your egg. If a mutation happens in either your sperm or your egg, that mutation will be in every cell of the offspring's body. And so that is how you pass a mutation on to your offspring, is having it made in your gamete. And so, what statement describes the evidence to support your choice in part A? It's that your offspring's genotypes, the genotypes of your offspring or the DNA of your offspring, are going to be determined by the gametes they inherit from their parents. The sperm and the egg that came together, that's the original set of DNA upon which every other piece of DNA in that organism's body are going to be copied from. And so if the gamete has a mutation, the new organism has a mutation. The other situation that you're going to be faced with on the state test will often be to identify mutations or to make mutations in DNA. And I'll tell you the biggest issue with figuring out these problems. So they'll give you a DNA and they'll tell you the amino acids that that DNA made. And notice in this one, it tells you what the changed amino acids are. But guys, the hardest part of answering this question is not knowing what the mRNA is. Because the mRNA is the link between the changes in the DNA and the amino acids that they get. So I always suggest the first thing you need to do is if you have DNA and you need amino acids, always write down your mRNA. That way, you can use your mRNA to locate amino acids, and you can work from amino acids to mRNA to DNA or from DNA to mRNA to amino acids. You can't go from DNA straight to this chart. You have to have mRNA. And 100% of the release questions that I have seen from the state of Ohio, they have the DNA and they tell you to find the amino acids or they list the amino acids and they leave that step for you to figure out, for you to write down. So in this case, in order for me to figure out which mutation was which, I really needed to know what the mRNA was so that I could figure out what change had to be made and whether it was an insertion or a deletion. The mutation was easy. The substitution, I'm sorry, was easy because in mutant number 2, there's only one change. CYS becomes TRP. And if it's only in one spot, it has to be a substitution. But the mutation 1 and the mutation 3, both of those affect multiple amino acids, which means they have to be either an insertion or deletion. And it actually took a lot of work for me to figure out that number 1 was an insertion and number 3 was a deletion. And so using this mRNA as a link between this codon chart and the amino acids to the DNA was a big, important step in how I solved this. All right, that's all I have for protein synthesis and mutations. Hopefully, you're working hard and reviewing, getting ready for your end of course exam. Let me know if you have any further questions